Dr. Zhongxi Yu, University of Illinois.
My research basically is in the field of nitrogen biogeochemistry. I was trained as an isotope biogeochemist. So specifically for this research project, we're going to look at the isotopes in conventional corn soybean rotation systems to look at the nitrogen cycling pathways and the intensity in those agricultural systems. So we will have basically two treatments. One is control. That means we will have 100% anhydrous ammonia application at 4. And the other one, the experimental treatment, that we will use split nitrogen application, 15% in spring and another 50% during the growth of corn. And for that experiment treatment, we also will have cover crops. So the goal of this research will, the first goal will be just look at the treatment effects on how nitrogen is cycled and how much nitrogen as nitrate will be lost in tail drainage. So a unique part of this project is that we will use isotopes to tell us how microbial activities is transforming nitrogen in agricultural soil and also lead to the loss of nitrate in tail drainage. So basically, my research look at different isotopes of nitrogen and oxygen. So basically, isotopes are the same chemical element but with different number of neutrons. So as a result, for example, for nitrogen isotopes, nitrogen 14 and nitrogen 15, they have different mass numbers just because of the extra number of neutron in nitrogen 15. And you can look at the natural abundance of those nitrogen isotopes. So we usually look at those two isotopes, oxygen 16 and oxygen 18. And the reason why we look at oxygen isotopes is nitrate has 3 oxygen atoms in it. So we are basically interested in to see the oxygen isotope abundance in nitrate. So although those rare isotopes are just tiny little bit in terms of their natural abundance, we actually can measure their relative abundance relative to these common isotopes in a very, very high precision and accuracy. So we plot oxygen and the nitrogen values together. And in this figure, you can see different sources have very unique isotope values. For example, you can see the manure and the septic waste have a very high nitrogen isotope values compared to other sources. Although we don't have much manure application right here, but if you look at the soil organic matter and also ammonium in fertilizer and rainwater, they are very different in terms of nitrogen isotopes. And also you can see right here is the nitrate fertilizer in UAN. So basically those nitrate, they got oxygen from atmosphere oxygen, the O2 in the atmosphere. So collectively, if you measure both oxygen and the nitrogen isotopes in nitrate, it can tell you a lot of process level information about where they're coming from and what kind of biogeochemical processes acting on those nitrate. But the ultimate goal, yes, is to use the N15 to differentiate different sources. Yeah, soil organic matter and apply the fertilizers. And also nitrogen recycled through the plant and soil systems. We'll sample tail drainage, we'll sample soil at different depths, and also we'll sample corn for biomass nitrogen. So they will give us basically a complete picture about how different processes change those isotope values in this system. So with this kind of comprehensive data set, we will look at first of all, look at their differences to see how, for example, tail drainage nitrate is different from nitrate in soils and also total nitrogen in crop. And that will give us a big picture about nitrogen cycling in the soil and plant system. So we will also measure, for example, soil organic nitrogen for its nitrogen isotopes, and fertilizers, both ammonia, anhydrous ammonia, and the UAN for their combined isotopic signature. So we will specifically looking at a variety of different nitrogen cycling processes, including mineralization, nitrification, immobilization, which is just the uptake of inorganic nitrogen back to the organic form. And in terms of nitrate, we'll be looking at leaching. Of course, we're looking at tail drainage. We will have a very good idea about how much nitrate is leached from the system. But we also will use isotopes to look at the denitrification process, which basically is convert nitrate to gaseous forms. So in addition to that, we will also look at basically the interactions between nitrogen and plants by looking at processes like uptake, nitrate uptake by corn, and also the return of biomass nitrogen back to soil. And in terms of soil being, we will use isotope-based method to look at how much nitrogen is actually fixed through the biological nitrogen fixation process. So this basically, you can see right here, each of those processes have those kind of red number associated with them. We call it isotope effect. That's just because for kind of those processes will change the isotope value. So when you look at the tail drainage nitrate, soil nitrate, you basically need to account for the transformation and alteration of isotopes in this system. So that's a very important part when it comes to we make our final conclusion about how much nitrogen is coming from, because potentially those processes can change the isotope value. But if we have this model, have this big picture, and do a very good job in measuring isotopes in those individual pools or reservoirs, that will give us a very, very unique and powerful opportunity to not only figure out what's going on in this system, but also let us know how much nitrogen is from soil and from fertilizers.